Hi all, Dr. Clark here again. And for this Gen Bio lecture, we're going to talk about prokaryotic, eukaryotic controls of transcription and translation. So remember that this diagram is showing a eukaryotic cell because you can see this nuclear envelope. Okay? But remember that in both cases, prokaryotic and eukaryotic organisms are going to go through transcription and translation, and it's nearly identical to each other. The one thing about prokaryotic transcription is that their entire genome or their entire chromosome is all genes, okay, back to back to back genes. Eukaryotic transcription, it's not back to back to genes. There's intron regions, there's non-coding regions, and that has to be processed. Now, that being said, there are ways at which transcription can be controlled. There are means by which RNA polymerase can be blocked, so it cannot come in and make a copy to make a messenger RNA strand. There are ways in eukaryotes that the SNRPs cannot bind and cannot remove introns, right, which will result in a faulty messenger RNA strand. Okay. There are also ways at which this can be controlled in translation. Okay? After the protein's even been translated, okay, it might fold incorrectly. Okay? It could be dissolved. Okay? Or during translation, you can have binding sites that can be blocked. Okay? So the translation process might not occur. Or the large subunit and the small subunit can never bind together because the small subunit or the small subunit's ribosomal RNA binding spot has been bound to and prevents the process from translation occurring. Okay, so I'm going to talk about these different ways in which prokaryotes and eukaryotes can control transcription and translation. This is important because, remember, amino acids are not like that easy to make. Okay, so it's not easy to just construct a bunch of amino acids. It's not easy to make messenger RNA strands and to go through the process of utilizing these proteins to make more proteins. And if you already have enough proteins, you have to have a means by which the system can be shut down. Okay? But what happens if you don't have enough proteins? You have to have a means by which the system can be initiated. And that's what we're going to talk about today. All right, so how do prokaryotes control transcription? It starts with prokaryotes. Okay, and like I said before, every cell in an organism, even if they're a single-celled organism, have to regulate when genes are used, when they're turned on, when they're turned off, okay, and when they're copied, when they're translated, and when they're transcribed. Okay. So part of this prokaryote system is because prokaryote systems are much simpler than eukaryote systems. They have regions that are similar to eukaryote systems where they have a promoter region. That promoter region is going to allow for RNA polymerase to bind to the DNA, okay, just like in eukaryotes. We talked about this before. Okay? But instead of regulating what happens to the messenger RNA after it's produced, most prokaryotes are going to regulate when the messenger RNA can be produced. Okay? So you'll have a promoter region, but you'll have a promoter region that is either turned on, so it can accept RNA polymerase, or the promoter region is turned off, so RNA polymerase cannot bind to the DNA site. Okay? And so those come in the form of either repressors, so they repress the promoter region, okay, and the region that is repressed is what we call the operator region. It therefore blocks access to the promoter region, so RNA polymerase cannot bind. Okay? Or you have activators. And so these activators will remove the operator or bind to the operator, so the operator can't have an effect on the promoter, which will allow for RNA polymerase to come in, bind to the promoter region, and make a copy of that gene that they're interested in, or the whole series of genes in the case of prokaryotes that's of interest. Okay? So again, if you remember back from previous lectures, prokaryotes are gene by gene by gene, and most of these genes occur in sequence. So you might have an entire biochemical pathway, all the proteins that are needed to maybe create another amino acid. Okay? They all occur in the same kind of block, in the same kind of region of the genome of a certain bacteria or prokaryote. So they're all copied at the exact same time. And they're all translated at the exact same time. So you transcribe them and translate them same time. So your entire biochemical pathway, protein, 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 they're all made at the same time. Okay? When this occurs, okay, you have to have a way at which either activators turn this on or repressors turn this off. Okay? Because you don't want to continuously make things that are not needed. So here you can see you have this promoter region and an operator region. Okay? If the repressor comes in, it sticks on the operator region. Okay? And RNA polymerase, it could bind to the promoter region, the PLAC, but it's not going to make it past that point. It will not copy these genes. So what you need is a way for that repressor to be bound up so the operator does not get blocked. Okay? And allolactose is one way. It's an inducer that blocks the repressor. That's one way you can do it. And you can block that repressor from ever binding to the operator. And this is called the lac operon. Okay? So lac would be that promoter region, and operon is going to be the operator region. So this is the lac operon region is either going to be repressed when the repressor is in spot, or induced when the repressor is being removed. Okay? If it's been removed, messenger RNA can be copied, so that DNA can be copied into messenger RNA, and the proteins in the series can be constructed. Okay? Pretty simple, but very efficient, because it reduces the amount of wasted product. If you already have all the proteins that you need in a biochemical pathway, then you don't want this to be turned on constantly. Okay? So the other piece of this is a lot of times the inducer or the repressor is part of this biochemical pathway or part of the end product. And maybe it's a substrate that comes off on one of these, and when there's a lot in solution, it can repress. Or when there's not a lot in solution, an inducer is constructed to stop the repressor. Okay? So there's a lot of things that can go on. So here's that lac operon, what we're talking about before, in the presence of glucose and lactose. Okay? So again, these are just carbohydrates, and this is a biochemical pathway going through the process of breaking down glucose or lactose in order to obtain energy. So if the operon is off, okay, so there is no cap, okay, you get plus glucose plus lactose. Okay, so positive. Okay. If the operon is off, okay, but the repressor is bound, okay, you can get the formation of glucose. Okay. If the cap is on and the repressor is found, Okay? RNA polymerase can't bind, and you get neither. Okay? And then if the cap is there but the repressor is gone, you can get the formation of lactose. So you're going from a ability to break down glucose or break down lactose, and it really has to do with whether there's a repressor present, whether there's a cap or the aperon cap is on, and this all determines what messenger RNA sequence is going to be synthesized, which will determine whether or not you can break down lactose or break down glucose. So here you can see kind of the same situation but in video form.
Many bacteria grow on glucose before they utilize other compounds, such as lactose, as a growth substrate when both are present in the medium. Transcription of the lactose operon is controlled by two regulatory proteins. The catabolite activator protein can bind to the activator binding site and facilitate transcription, and the lac repressor protein can bind to the operator site and block transcription. The catabolite activator protein can bind to the activator binding site only if cyclic AMP is bound to it. When glucose is present, the concentration of cyclic AMP in the cell is low. RNA polymerase cannot bind efficiently to the promoter, and transcription does not occur unless the catabolite binding protein is bound to the activator binding site. Therefore, transcription of the lac operon does not occur in the presence of glucose. In addition, if there is no lactose in the medium, the lac repressor binds to the operator site, and this blocks transcription. When both glucose and lactose are present, there is insufficient cyclic AMP to bind to the catabolite activator protein, which therefore cannot bind to the activator binding site. Without the bound activator protein, RNA polymerase cannot bind to the promoter and initiate transcription, even though lactose binds to the repressor and prevents it from binding to the operator site. When neither glucose nor lactose is present, the concentration of cyclic AMP is high, and the catabolite activator protein is bound to the activator binding site. RNA polymerase can bind to the promoter, but it is blocked by the repressor on the operator site. When glucose is absent and lactose is present in the medium, cyclic AMP is present and binds to the catabolite activator protein, which in turn binds to the activator binding site and facilitates transcription. Lactose binds to the repressor and prevents it from binding to the operator site. RNA polymerase can now bind to the promoter and carry out transcription.
Okay, so from there you can see how the lac operon works in the case of prokaryotes, some prokaryotes, okay? In that organisms have the ability to switch from growing on glucose to growing on lactose. Okay? And they do so by either promoting a region or repressing a region. Okay? And this is very, very simplistic form, okay, of transcription control. You're going to see that as we move into eukaryotes, we have similar controls like this, but we also have much more complex controls where we're not just talking about the presence of, two sugar molecules. Right? We're talking about all kinds of things. We're talking about end products coming back into the system to repress, and lots of different situations. But nonetheless, it's the same idea. There are repressors and activators, and depending on the situation, some things will be repressed to prevent, a messenger RNA being formed, and some will be activated to promote the messenger RNA being formed.
In the bacterium Escherichia coli, a group of 5 genes code for enzymes required to synthesize the amino acid tryptophan. All 5 genes are transcribed together as a unit called an operon. An operon is a group of genes that is under the control of a single operator site. A regulatory protein called a repressor can bind to the operator site and prevent transcription. When tryptophan is lacking in the environment, the repressor is made, but it is unable to bind to the DNA and block transcription. RNA polymerase binds to the promoter site and then proceeds down the DNA, transcribing the genes for the tryptophan biosynthesis enzymes. When tryptophan is present in the environment, the organism no longer needs to make tryptophan. Tryptophan binds to the repressor and activates it. The activated repressor now binds to the operator located within the tryptophan promoter and blocks transcription.
Okay, so this is one of those negative feedback loops. And remember when we were talking about biochemical pathways, and I said, at the end of a biochemical pathway, you often get this product that comes back to the protein and closes off the activation site. It's the same premise, except for here, we're talking about not copying DNA and not making the messenger RNA so you can make the protein tryptophan. Okay? And so tryptophan is utilized in its own repressing of the production of tryptophan. Okay? So you have too much in the solution, okay? It's going to block that operator region, so RNA polymerase cannot bind and you cannot form the protein tryptophan. Okay? All right, so transcription controls in eukaryotes can be very similar to prokaryotes. But remember that we're not talking about an entire organism, okay? And when we're talking about prokaryotes, we're talking about the entire organism. They only have one chromosome, and they're only one cell big, typically. So when you regulate that, you're regulating the entire organism. Okay? Now, when we talk about gene regulation in eukaryotes, we can talk about genes being turned on or turned off, but you have to regulate this not just in a single cell. You have to regulate this in all the cells that might produce, a certain enzyme, a certain biochemical pathway, all the cells that might, produce insulin or a certain amino acid, right? Because at that rate, then you're shutting down the entire system. So you don't just shut down one portion of your body that's producing insulin while another portion is still producing it. Okay? You have to shut down the whole system. Okay? Ways that you can do this is by changing how the DNA is regulated through the packaging of chromosomes. Okay? So chromosomes are wrapped around histones to make nucleosomes. We've talked about this before. Well, there are ways at which you can methanize that chromosome to prevent the expression of a certain gene. Okay, so if you change the availability of chromatin or availability of the DNA for transcription, that's one way that you can affect the entire organism. Okay? You can affect every single cell in the organism by changing how available the DNA is to transcribe. If it's bound up by proteins, then RNA polymerase cannot get in there into the DNA and unravel it and make a single copy. So here you can see DNA wrapped around histones, okay? And if it's tightly coiled around histones, there's no way for RNA polymerase to come in here and make a copy of a certain gene. This is one way that we can prevent, right, the overexpression of genes in eukaryotes. So you can do this in a couple ways. Okay? You can increase the condensation or the condensing of the chromatin. Okay? So how condensed it is. So we can do this by adding material that increases the stickiness of chromatin. Okay. We can methylize the DNA, okay, which basically means that you put a coating over it that turns off the genes, and the genes stay off till that methylation is removed. Okay, if it can be removed. In some cases it can't. Okay. And you can use other co-activators, proteins that can come in and methylate histones, okay, which makes it so the chromatin cannot coil around the histones. So basically you're coating the protein so DNA can't wrap around it, so now you have access to the DNA. So there are ways to turn genes on by removing the histones, and there are ways to turn genes off by ensuring that the DNA is going to wrap around histones and stay wrapped around histones. Okay, now there are also co-repressors, and co-activators and co-repressors will often act upon histones. So how likely or how tightly wrapped is the chromatin around histones, around these proteins, or how loosely, or the binding properties. So in some cases they're wrapped really tight and no RNA polymerase could get in there, and in other cases there's no wrap at all. There's a disassociation with the histone and the DNA is open for replication. Right? It really just depends on whether you're activating or you're repressing. Now, there are other ways in which this can occur. Okay? Transcription, because it's more complex in eukaryotes, okay, you have a bunch of other proteins involved. Okay? So you don't just have a promoter region and an operator region, but you have other regions that are going on. You have regions that promote, okay, but regions that are what we call co-promoters. And so they might occur before the promoter region, which allow for binding proteins to come in. Okay. And so you can get what we call basal transcription factors, okay, which are going to initiate and recruit RNA polymerase to the promoter region. Right? So we talked about this when we were talking about the polymerase, RNA polymerase coming in. We said that there are these specific factors or these single binding factors that come in and allow for RNA polymerase to bind to the promoter region. Excuse me. Those are things like basal transcription factors or specific transcription factors, okay, that affect the rate at which RNA polymerase binds, that affect the rate at which the gene can be transcribed, so it can slow down the transcription process. It can speed up the transcription process. It can determine whether or not the transcription process can occur at all. Okay? And so it's much more complex than, say, the prokaryote level. But nonetheless, it works the exact same premise as we talked about before when we were talking about negative feedbacks, positive feedbacks. Okay? You often have an activator or a repressor. These bind at different regions and allow for different complexes to occur. RNA polymerase to bind, RNA polymerase not to bind. Okay? So here you can see this promoter region. Okay? It's a little different in eukaryotes. It's typically a lot bigger, and then you have a what we call a front end. This is the Tata box. Okay? You have this front end that has a whole series of proteins that are associated with. Okay? In some cases, every single one of these proteins has to be associated in order for RNA polymerase to bind. All it takes is one repressor molecule to bind up one of these proteins, and RNA polymerase cannot bind. Now you can have what we call enhancers, okay? And these are just like activators. Okay? So they enhance or activate the transcription process. They can be bound up or they can be associated with what we call co-activators, okay, or mediator proteins or mediation proteins that can either allow for transcription to occur at a slow rate or a fast rate, or in some cases not at all. There's a whole series, and to tell you the truth, some of these messenger RNA sequences, some of these genes, we actually don't know how many enhancers are involved with it or how many co-activators are involved with it. When we figure this out, we can do things, potentially, with modern medicine to add a co-activator. So maybe you need to produce more insulin, or maybe you need to produce more of a certain amino acid. Okay? So you could add a co-activator and stimulate that messenger RNA completion. Okay? On the flip side, you can also shut down systems. Okay? And so unlike prokaryotes, where it might be fairly simple and you might have a repressor and an activator, in eukaryotic systems, depending on the gene, depending on the organism, you can have multiple co-activators, multiple enhancers that are going to activate the process of transcription, or you can have the opposite true. Multiple co-repressors that stop the process from occurring. So here you can see just kind of an example of how this enhancer works. So you have an activator that's going to bind to an enhancer sequence. Okay? And then you can have a transcription factor that comes in that's going to bind to the promoter region. By doing that, it causes a hairpin loop in one strand of the DNA, which allows for these two, the transcription factor and the activator, to change shape, to interact with one another, to provide a binding spot for the RNA polymerase to bind to the promoter region and start messenger RNA synthesis. Okay? So there are a lot of things that can take effect, and just one minor change and say, what is the transcription factor? Okay? Is the transcription factor there? Is the transcription factor have a repressor on it? Okay, if it does, it won't bind to the promoter region and it will stop the entire process from occurring. So here you can see, this is a kind of a legitimate system for transcription. Okay, I talked about the TATA box. Okay, this is normally that right in front of the promoter region, you'll have a region which has a number of binding proteins. Okay, these are called basal factors. Okay, they bind up to the TATA box. Okay, they can have co-activators that are bound to it. They can have activators that are bound to it, which can cause these hairpin loops, and you can get numerous activators. Okay, and all of it's needed in order for RNA polymerase to bind to the promoter region and start the messenger RNA sequence. Okay? This is a great system. It can be extremely complex, but it's a great system in the fact that there's all these feedbacks. And as eukaryotes, we rarely waste material, especially in process of making messenger RNA to make proteins. We're not wasting material. We're not wasting amino acids. We're not making unnecessary messenger RNAs. We're using a numerous amount of activators and enhancers, co-activators, all these basal binding factors, TATA box, which is going to be part of the promoter region, all to initiate RNA polymerase binding, okay? And then that can copy the DNA. And I told you before, when we were talking about DNA synthesis, that I'd talk about polymerase 3, polymerase 1, right? And I talked about those when we were talking about synthesis. And I said I'd come back and talk about polymerase 2. And polymerase 2 really has to do with the copying of small segments of DNA. Okay? So RNA polymerase 2 is the enzyme that's going to lay down RNA nucleotides to form that messenger RNA strand. Okay? And so that's where the 2 comes in. It's normally not associated with copying the entire genome, but rather small segments, small genes that are associated with transcription and translation. All right, so there are other ways at which this can be controlled. You can control it at the RNA level. So once the material has been transcribed, okay, now you can alter the way at which that material is expressed. Okay. You can do that by developing what we call double-stranded RNA. So sometimes those messenger RNA strands will double strand. They'll go double helix, okay, and you'll get a gene silencing. So in other words, you made the messenger RNA, but sometimes either another messenger RNA will lie on top of it and block the translation of that gene. Okay, sometimes it can just fold over on itself and form a hairpin loop and go double helix. In these cases, we call that gene silencing. It's turning the gene off, even though the RNA has been copied, it has been made. Okay, so here you can see how that works. So you can see that in this case, the RNA formed what we call a hairpin loop. It went double helix. Okay, it became double-stranded. And there are ways at which you can change this interference. Okay? We have means by which we can actually translate this later on. Okay? So at first it's inhibited. It's like shut off. It's shut off the messenger RNA. Okay? But you can have situations where you get siRNAs, which will come in and they'll dice up the double-stranded RNA, and then they'll separate. They'll unwind that double-stranded DNA, so you have a single-stranded messenger RNA. Okay? In some cases, that messenger RNA is allowed to be translated. Okay? In other cases, it's destroyed because it was damaged, it was cut, it's not the original size, okay? And it just depends on how this interference worked. Okay? But it's really in regards to your messenger RNA went double-stranded. Okay? It formed its own hairpin loop, okay? And then we have mechanisms to destroy that. Okay? We can use dicers and things like that, which will destroy the original sequence. Okay? And then you can just make a new transcription of the gene afterwards if it's needed. Okay? All right, there's other ways. Okay? Again, eukaryotes, because we're so complex, there are lots of ways at which we can regulate gene expression. I already talked about chromatin structure. Okay? So whether or not your chromatin are bound to the histones or not bound to the histones, this will prevent transcription from occurring or promote transcription to occur. Okay? The initiation of transcription. Okay? The promoter regions, the Tata box, the enhancers, the repressors that can go on, okay, can either initiate transcription or not. You can have alternated splicing. So you can have the messenger RNA that is spliced incorrectly. Okay? So that messenger RNA is terminated. The introns could be left in, which means the messenger RNA is terminated. It can, you can remove all the introns and splice the exons differently, which ultimately would allow you to express the gene in a different form. Okay? Another way in which gene expression can be changed. RNA can interfere. So you can have RNA double-stranded, okay, RNA coming in and binding up to the messenger RNA to prevent it from being expressed or translated. There's lots of situations. Translation proteins. Right? In some situations, you have binding proteins that are associated with the small subunit and the large subunit of the ribosome. And if those translation proteins are not available, the subunit, the small subunit and the large subunit of the ribosome will come together and they'll stick for a little bit, but then they disassociate before the protein has been created. Okay? And then there's lots of situations after translation has occurred. So you already made this amino acid strain. Okay? Now we have to initiate the wrapping, either the forming of, say, an alpha helix, a beta sheet, okay, spirals, all these things. In each one of those steps, you can have modifications. You can have proteins that will bind incorrectly, and it'll cause for a misshapen protein, and the product is terminated, it is dissolved, okay? And you start the process all over again, or the process has been terminated because you don't need any more of that product. Okay, so again, like I said before, there's lots of situations that eukaryotes can do this. Okay, chromatin, how the DNA is packed. Okay, RNA transcription, whether or not the promoter region is there, whether or not Tau box is blocked, all kinds of things. How the introns are being removed, exons are being put back together. Whether or not that messenger RNA forms a loop on itself, forms a hairpin loop when it moves into solution, okay, and gets cut into small pieces. Whether or not there's proteins to allow for the ribosomes to associate with each other, and then again, at the end, after the amino acid chain has been formed, does it actually form the right protein? Do the proteins actually form the right shape? Lots of things to think about. Okay, we're going to watch a little video about some of these gene expressions and how they work.
Most of the control of gene expression in eukaryotes is achieved by regulating the frequency of transcription initiation. Various regulatory proteins bind to DNA and affect the ability of RNA polymerase to bind to the promoter site and carry out transcription. Efficient binding of the RNA polymerase increases the rate of transcription initiation. Gene expression can also be controlled by altering the splicing of exons in eukaryotes. For example, the faster messenger RNA is processed, the greater the amount of gene product. Gene expression can also be regulated by controlling access to or efficiency of transport channels. The passage of an mRNA transcript across the nuclear membrane is an active process requiring that the transcript be recognized by receptors lining the interior of the pores. Many enzymes degrade messenger RNA. Gene expression can be regulated by modulating the degree to which the messenger RNA is degraded. Many different proteins and amino acids take part in the translation process. The rate of gene expression can be regulated by speeding up or slowing down protein synthesis. The availability of any of the proteins or amino acids can alter the rate of the protein synthesis. Phosphorylation or other chemical modifications can alter the activity of a protein after it is produced. This is referred to as post-translational modification.
Okay, so from there you can see some things that I didn't even talk about, and that's things like the nuclear pores of the nuclear envelope not allowing for messenger RNA to come out. Okay, there's also enzymes that will cut DNA as it's floating around in the cytoplasm. If they encounter DNA, they'll destroy that DNA. And this is a way to ensure that after a messenger RNA has been made, okay, and it's sitting in the cytoplasm, that it doesn't just sit there and keep making the exact same protein over and over and over again. If there's too much of that protein, then that material is going to be cut up, it's going to be bound up, or there won't be enough amino acids in solution to form that protein. There's lots of ways at which gene expression can be regulated in eukaryotes. Okay, so with that being said, we're going to kind of switch gears a little bit, okay? And we're going to start talking about evolution. Okay? Now, we talked about how DNA is copied, how mistakes are made, how things are regulated, on and on and on. Now we're going to talk about, well, what happens when those mutations come to fruition, when those mutations are actually shown in a phenotype. How does nature act upon those mutations? How does natural selection, not just natural selection, because that's only one mechanism of evolution, but how does natural selection, sexual selection, artificial selection, gene flow, genetic drift, how do these concepts, these evolutionary concepts, act when there's a mutation in the genetic code? Okay? So next time we're going to talk evolution.